Assalamu alaikum, everyone. Can you hear me? I think we can start. So the chat box is open for you guys if you have any questions. Just to make sure you guys can hear me. Okay. So I think we can start. Let's just make sure that we are recording. Okay. Okay, Assalamu alaikum everyone. I'm Sabehat Ali, and today we'll be reviewing nucleic acid chapter. To be honest, this is one of the chapters that is mostly memorizing based. It's not like understanding based. However, understanding some certain concept will help you just to memorize it. Some concepts are actually more understanding than memorizing. So this session will be, I divided the session basically for 4 categories or 4 subcategories, and we're gonna discuss each one of them alone. So let's start by this question. You can answer in the chat box. Okay, so most of you guys said A, which is correct. Yes, it is genome. What about this question? Okay, I see C and A. What else? It is A. Okay, so the first concept that we're going to talk about is the DNA. So basically, guys, these are some definitions that you need to be familiar with. For example, what is DNA? What is genome? Was the first question, right? Genome, it's the complete DNA based sequence. This is genome. What about transcriptome? So basically, sorry. Okay. Transcriptome is basically it's the complete set of RNA. DNA synthesis, we call it replication, while transcription is RNA synthesis, right? So this is just pure memorization, nothing to be explained. Also, we have metabolome, proteome. Proteome is the entire set of protein within a cell. Metabolome, it's the low molecular weight of metabolites used within the cell during gene expression, and so on. So what about this question? Is it hard, guys? Nobody answered it. Okay. I can see so many C's. It is it is C. Yes. It is C. So what is actually central dogma? Central dogma, think about it, is just a sequence of how are we making the protein. So we start from DNA by replicating the DNA, making let's say another copy of the DNA, then transcribing this DNA, so it will become RNA, and then we translate it to proteins. So this is like the sequence that we go with. So we start with DNA again, moving to RNA, and then we translate to protein. This is the sequence. Now, some enzymes, or let's say some viruses, they can do what the thing is that they can invade the host cell, and then what they can do is they take the RNA and then they reverse it. So as I said, we go from DNA to RNA, right? So the thing that with these viruses, that they can go from RNA to DNA. They have a special enzyme called reverse transcriptase to do that. So just know it. usually found in most of the viruses, especially in HIV virus, which causes AIDS. So it can go from RNA to DNA. However, the normal sequence is from DNA to RNA to proteins. Now here, as you can see, there is like parent and daughter. It's basically parent then daughter DNA. So just don't worry about it. They won't ask you about. So what about this? Okay, most of you guys answered A. It is A, correct. We'll go back to it. What about this question? A again, correct. Moving to this. Yes, it is B. What about this? Yes, it is D. I think this is the last question. Yes, it is A. Okay. So basically this photo, the one in the left is from your book, and the other one I stole it from my year's one book. So basically here, what do we have is the DNA structure, right? So let's start by this photo, which have the nucleotide on it. This is the nucleotide. So what is the structure of DNA? Basically, we have a deoxyribose, which is a reduced sugar, and then we have a phosphate, which makes the backbone, and then we have a base, which is the glycosidic, which is one of the nitrogenous bases. Now, the bond between the phosphate and the phosphate, and which we call it like basically the backbone, which is the phosphate and the deoxyribose, is the phosphodiester bond. And the bond between the ribose and the base is a glycosidic bond. Make sure that you know these guys. And the other bond between the base pairs is hydrogen bond. Now we have four types of base pairs in the DNA, which are adenine, guanine, thymine, and cytosine. The two adenine and guanine are purine, and thymine, cytosine are pyrimidines. Now, the thing is that each pyrimidine will be bound to purine. Now, why we don't bound purine to pyrimidine? I think, can I draw here? Yes, I can. So basically, let's suppose that we have here, it's too laggy. Let's suppose that we have, this is a pyrimidine. You know, pyrimidine have one ring and purine have two rings, right? And then we have another pyrimidine here. And then we have, okay. And then we have purine. I don't know how to draw, guys, just, and then a purine here. And then again, pyrimidine, pyrimidine. Now, the thing is, the distance between purine, between pyrimidine will be close, like far, because they are like one ring, and the distance between the two pyrimidine, the two purines will be shorter. That will make the DNA be in this shape, which is not straight as we know. DNA has this shape, and then it will start like to twist to have this helical shape, right? So in case of we have purine bound to purine, this what will happen. So in order just to make the distance between base pair like a stable DNA, we need to have a purine bound to pyrimidine. And then we have a ratio, which is by the Shurooq rule, which says that thymine should equal to adenine and guanine should equal to cytosine. Why? Because adenine bound to thymine and guanine bound to cytosine, right? One more thing is that between adenine and thymine, we have two bonds, two hydrogen bonds, and between guanine and cytosine, we have how many bonds? Three bonds. So if we went back to these questions, starting from this one, so this question states that there is a heat and there is denaturation of the DNA. So which one would you think will, denature the first? Is the one that has two hydrogen bond instead of three hydrogen bond. So basically, guys, just search for the sequence that has more AT pair, because more AT pair means that we have two H bonds instead of three H bonds, which basically means that these like sequence will degenerates faster. What about this question? So as I said, we have purine and pyrimidines. So as you can see, uracil, which is found in RNA, basically cytosine and thymine, they are all pyrimidines. The only one that is purine is AMP, which is adenosine monophosphate, as you guys answered. And a nucleoside, basically nucleoside is the deoxyribose plus the base pair without the phosphate. So it's nucleotide minus the phosphate, right? One more thing is the Shurooq rule, as we said, that it is adenine will equal to thymine and cytosine will equal to guanine, as they are base pairing each other. This question we answered, and this is just, guys, a free mark. Just know how to read the sequence of DNA. It is highly important concept. يعني تقريبا in almost بين كل امتحان وامتحان they get it. So just make sure that you know how to read the concept really well, and how to know how to read the DNA concept and the DNA sequence and the RNA. And for example, if I ask you for DNA and then I said the sequence of RNA, the T will be U, right? On such a question. And I think in that way, this answer will be like U, uh, sorry, it will be yes, A, U, G, C, and then again A, right? So know how to read like from DNA to RNA, from DNA to DNA. Okay, so I'll just stop here to see if you guys have any question, and then we'll proceed. So if you have any question, guys, you can just raise your hands. Okay, someone was asking about the glycosidic bond. Sorry, I raised my hand by mistake. Someone was asking about the glycosidic bond. The glycosidic bond is between the sugar base and the sugar base and the nitrogenous base. I think you took chapter 17, which is about sugars and carbohydrates. So whenever you have a sugar, which is ribose is one of the sugar, right? So the sugar will be bound to a nitrogenous base, which is thymine, guanine, cytosine, or whatever. So this bond between both of them will be a glycosidic bond. I'll just go back to that slide. Here, the glycosidic bond between the base pair and the deoxyribose. Okay, so where is the hydrogen bond between the rest of them? So basically, the hydrogen bond, as you can see here, it's between guanine, cytosine, thymine, and adenine. If this is your question, يعني, I hope I got your question right. Is that what you mean, يعني, or something else? Okay, nice. So we can proceed, right, guys? Or you have any other question? Okay, no one is raising their hands. We can proceed. Okay, genetic changes. Let's start by this question. Okay, someone asked if the phosphodiester bond should be between the nucleotides. It is between the nucleotide. It is in the backbone. And this photo, because it's just a single nucleotide, it's not showing well. Here, we just wanted to highlight that, no, this here is the bond exactly. But as you said, it's going to be between the nucleotides. So it's يعني, one phosphate will be bound to two nucleotides. That's why we call it phosphodiester. Di means two, basically. So in the question I gave you, you answered transition mutation, which is totally correct. Next. So some of you guys answered C and some answered A. The correct answer is actually A. It is alkylating agent or ionizing radiation. We'll go back to this question. What about this one? Transition mutation of cytosine will give us basically thymine. And if we are talking about DNA, basically, if we are talking about RNA, it would be uracil. Okay, so what is transition mutation and what is transversion mutation? Basically, transition mutation, that means that we have the same ring, which is the purine ring or pyrimidine ring. So it is mutation between the same group of rings. It's either purine to purine or pyrimidine to pyrimidine. So it's either for transition mutation, it will be from guanine to adenine. From guanine to adenine. What about transversion mutation? Transversion mutation will be from thymine to cytosine. Sorry, from thymine to adenine, or from thymine to guanine, or from adenine to cytosine. Why? Because adenine is it's a purine, and cytosine, for example, is a pyrimidine. So if we change from purine to pyrimidine, that's a transversion. If we change from purine to purine, that's a transition mutation. Let's go back to the first question that you guys answered. Usually, changing from purine to purine or from pyrimidine to pyrimidine is by the cause of deamination reaction, which will be followed by frame shift, or let's say, yeah, which would be not frame shift, tautomerization reaction. So basically, these two will come together. So basically, these two will come together, which is deamination reaction and tautomerization reaction. So let's see this question. And this question, they said that which one of the following is the common cause of by mutation that is resulted by a change from purine to pyrimidine? From purine to pyrimidine, mostly it is caused by alkylating agents or ionizing radiations. As I said, deamination or tautomerization will be for what? Will be for transition mutation, which is this question, right? Because we said from pyrimidine to pyrimidine and from purine to purine. For transition mutation of cytosine, as I said, transition means from purine to purine or from pyrimidine to pyrimidine. Since cytosine is a pyrimidine, that means that we need to find another pyrimidine in the choices, which is thymine in case of DNA or uracil in case of RNA. Okay, moving to this question. So yes, this question, the answer of it is C, which is nonsense. So why? Because they said that in codon number 91, so we look at this one. It was changed by UAA instead of UAG. Now UAA is a stop codon, which makes the answer nonsense. Why? Because we stopped the protein formation before before the real stop codon, صح? So it doesn't make sense. We said that it's nonsense and it doesn't make sense to stop the protein synthesis ونحن لسه أصلا شنو ما وصلنا the real stop codon. So this is a nonsense mutation. What about this one? So someone is asking about if we need to know the stop codons or not. Now, the thing is, for your, I saw your PowerPoint, guys. It didn't have the stop codon, neither the start codon. But just know it. Just know it, يعني. What about this question, guys? It is a missense. Why? Because we missed the read of the real of or the actual amino acid. So the actual amino acid was glycine, and we changed it by serine, so we missed the real or we missed the reading. So it's a missense mutation, correct? Now let's just sum up quickly. So this is the non-mutated DNA or RNA. Let's just look at the RNA or DNA, it doesn't matter. So for the first one, it says AAG and then it becomes AAA. So we changed G to A, and then it gave us lysine again. So we say that this mutation is silent because we have the same amino acid. What if we change this A to U or this T to A? Now we change it to stop codon, which doesn't make sense. So we call it nonsense, which will give us stop codon in the middle of synthesizing the proteins. What if we change it to, for example, another amino acid? This is a missense because we missed reading the correct amino acid. So here, as you can see, it's just conservative and non-conservative. I don't think you took it, guys, but just for your information, conservative, it means that we change to the same group of amino acid. Non-conservative means that we change to different group of amino acid. So no, guys, we took it in protein chapter. So basically, lysine is a basic amino acid, and then arginine is another basic amino acid. So we say, this mutation is conservative. Threonine is a polar, lysine is a basic, so they are in different groups. So it's non-conservative. There is an example in your book, which is sickle cell anemia, if I'm not mistaken, which is between valine and glutamate. Okay, moving to mutations. So basically, we have silent mutation, nonsense mutation, and missense mutation that I just mentioned. But here there are two more examples that I need to talk about, which is frame shift mutation. Frame shift mutation is basically caused by indels, which is N insertion and dels deletion. So basically, it's whenever we add a single nucleotide, okay, to the sequence. And how do we know if this indel is frame shift or not? We basically divide by 3. If it was divisible by 3, that means that it's not frame shift. If it wasn't, that means that it is a frame shift mutation. What about repeat expansion mutation? It's a type of a mutation, usually insertion mutation, which will cause the DNA basically to repeat a certain sequence or a small nucleotide sequence, and that might cause some diseases or some other complications. Now, what about this question? D, translocation, or D for what? All of you guys think no, it's D. No other answers. A. Yes, it is A. It is A. Okay. So basically, this is one of the easiest concept. Again, guys, just go through it because it's again a free mark. So duplication is basically when we have a small sequence of DNA duplicated twice, as you can see here. B is duplicated twice. Deletion is basically when we have a delete or a missing sequence in the other chromosome. What about inversion? Inversion is the one that I just asked you about. It's basically when we have a chromosome. Now, usually chromosome is split into two sides, right? Let's name this a P side and this a Q side, the down one, Q side. Now, if the DNA or the special sequence that we have was in the P side and it becomes in the Q side, this is inversion because we invert its site, right, or its location. Now, another type of mutation is translocation mutation, which will usually be associated with two chromosomes instead of one. And basically, it's when we dislocate or we change the location of a specific DNA from one chromosome to another. And for example, there is a disease called Down syndrome. It is caused by translocation mutation, where chromosome 13, it was 13 or 14, chromosome 21 is translocated. So, guys, do you have any question before moving to the third part? Okay, I think we can proceed. Okay, so again, another question. C, yes, it is C. So basically, these are the three DNA structures: B form, A form, and Z form. So B form is basically it's the first form that was found. It is that represent like in a DNA in the human condition, which represent, sodium and salt being their normal DNA. A form is basically when it's a bit dehydrated, so it start like to have a little bit of shrinkage. This is the A form. So usually there is water, molecules bound to the DNA itself. So the water molecules bound to this DNA will little bit decrease. And the Z form, which is usually caused by, torsion, and it have unknown also causes. It is we will have a complete twist to the other side, which is the left side. This is another photo that I put just to see how the Z form has like a different conformation than the B and A form. So what about this question? Okay, so it is A, it's not B, and I'll explain why. What about this question? Basic and DNA, correct. Yes, it is A. Okay, let's talk about prokaryote a bit. So basically, prokaryotes, which are like bacteria, such as E. coli, um, they will have a chromosome that is circular. Our chromosome is basically linear, it's not circular. So they will have a circular DNA, which is looped and coiled. Now, what is special about these prokaryotes that they are like, looped through a nucleoid associated protein. They would be like highly supercoiled and then wrapped into these proteins, just to save space. And because, DNA are like highly huge and they are pretty long, so we need to compress them. So we do the supercoiling. And one more thing, that they are positive in charge. Why do we need them to be positive in charge? Because basically, DNA, as we said, we have a backbone of phosphodiester bond, right? Now, this phosphate will have a negative charge. Phosphates are negative charge. So we will need something to be positive charge to just inhibit or prevent the repulsion. Negative, negative will repel each other. So we need something that is positive charge just to neutralize these charges. Another thing that we're going to add is polyamines. We'll bind them to these negative charged in the DNA backbone just to overcome the charge repulsion. What about eukaryotes? Eukaryotes are basically, we don't have this. One more thing, guys, nucleosome and nucleoid are not the same. Just don't mix up with the two terms. Nucleoid are for prokaryotes, and we have nucleosome. It's basically histones wrapped to DNA. So basically, eukaryotes possess an um, let's say in eukaryotes we have single linear DNA, as the question mentioned. Now I know some of you guys said B because genomes are multiple. It's not here, sorry. This question, multiple linear double helix. Basically, some of you guys said multiple because of the genome. I'll explain this later when we finish the PowerPoint. But you need to know that DNA molecules in the chromosomes, they are single linear. Okay. So basically, we would have a DNA and then we will wrap it to histones. Now, I didn't mention histones here alone. I didn't talk much about it. We'll talk about it by the end of this meeting again. So it would be wrapped to histones, usually 8 histones, and this will create the nucleosome. And then this nucleosome will come together as a condensed fiber. Now, all these condensed fiber, when they come together, they will make the chromatin, which is basically then makes the chromosomes. Now, as you guys said, histones are positively charged because, again, we need to neutralize the backbone, right? So, guys, do you have any question before moving to the last part? Okay, I think we can continue. Yes, it is A, which is basically OH groups of RNA confers extra reactivity because, as we said, DNA, it's deoxyribose, which means that it lacks OH in the second carbon. So when we have this OH, it means that better reactivity basically. The single stranded structure allows folding into complex shapes. That's correct, such as tRNA and rRNA. They are more complex than the DNA itself. So let's talk about RNA a bit. So basically, they are sugar, ribose sugar instead of deoxyribose sugar, as we said. That means that they have an reactive OH in their second carbon. So, as we also mentioned, we have uracil instead of thymine, and also we have it is a single strand. It's not a double strand. However, we can have again a base pairing between them because RNA and DNA both of them are like too flexible, so they can just arrange in any shape and they can do like base pairing as well. So one more thing that we don't follow here Shadev's rule because RNA molecules, as we said, they are single-stranded RNA. They are not double. So we can't say, this equals to this. And one more thing that you guys took in chapter, the enzymes chapter, that most enzymes are proteins. And here we have a small exception, which is ribozymes, which is basically another enzyme. However, it is made of RNA. It is a nucleic acid which have, yes, catalytic activity. What about this question? Yes, yes, it is ribosomal RNA. It is the most abundant. What about this question? Yes, it is C. Somebody asked which one is more soluble of them. Do you mean like the DNA and RNA ولا prokaryote eukaryote صراحة because it's been like كثير رسائل till that message. A, what does A says? Yes, it is A. So, okay, let's talk about messenger RNA, ribosomal RNA, and transfer RNA a bit. So, for transfer RNA, it is basically the RNA that does the one that transfer amino acid and makes the peptidal bond. So, as the question said, the 3' CCA sequence of the tRNA is the site that will be covalently bound to the conjugate amino acid. This is important, guys, just know it. So, basically, it is a single-stranded RNA that has the codon. If it has the codon, what will have the anticodon? Basically, messenger RNA will have the anticodon, right? So, no, wait, I'm sorry. I think it has the anticodon and the messenger RNA has the codon. I'm sorry. So, as I said, so the transfer RNA will have the codon, and then the messenger RNA will have the codon to match between both of them, and then they will bring according to this matching. For example, if it was AUG, that means that it will be methionine and etc. So it will bring the methionine amino acid after it forms like a long thread of amino acid. This amino acid will be folded, and then it will be like a special protein. Ribosomal RNA is basically one of the most abundant RNA. Just guys know which one is the least abundant, which one is the most abundant. We have two form of it. The eukaryotic one has two subunit. The small subunit, which is the 40S, and the big subunit is the 60S. If they are combined together, we call them 80S subunit. Same goes for the prokaryote. However, the small subunit in prokaryote, we call it 30S. The big subunit, we call it 50S. Together, we call them 70S. Now, what about messenger RNA? Messenger RNA is basically almost 5%, which means that it is the least between all of them that will have the codon on it, which carries the genetic information that we bought from the DNA after the transcription, right? So this messenger RNA also have like, whenever the base sequence that we have in the messenger RNA are able to synthesize a protein, we call them open reading frame. Polycentric, yes, polykaryotics are polycentric. What about this one? Yes, it is D. It is none of them. So basically, we talked about the open reading frame, right? So now the open reading frame will have an initiator and then a terminator code, which is AUG at the beginning, for example, and then at the end, we'll have UAA, for example, which is a stop codon, right? Someone was saying, what should the answer be for I think for this one, what codes for GNA product? As we said, open reading frame codes for GNA for DNA product for a protein. However, introns they don't code. They don't code. We just cut them. 5' and 3'. I'll just explain them now. The 5' UTR and then the untranslated region يعني من الاسم untranslated region basically they will stay on the beginning and the end of the open reading frame or of the like area that was going to be translated into protein. um what is going to be translated into protein, as we said, it is the open reading frame. Now there is something that you guys also you need to know, that for prokaryotes and the same like in the same messenger RNA, they can synthesize more than one protein. That's why we call them polycistronic. Why? Because they can synthesize or can make one protein, more than one protein from the same messenger RNA. However, for us, messenger RNA codes only for a single polypeptide. So we call it monocistronic. As this question said, where is it? This one. Not this one, this one. So let's just go over the هذا. If it's synthesized with introns, we said no, can only be monocentric. No, because they can be polycentric. Now, what about this question? I think this is the last one in the slides. snRNA. Correct. It is snRNA. So snRNA, it's a non-coding RNA. So it's basically, just to clarify, non-coding RNA are there in the, or non-coding DNA are there in the prokaryotes and eukaryotes, but the percentage is higher, more higher in the eukaryotes. Now here for the RNA non-coding or non-coding RNA, most their function is usually to just to regulate and um, yeah, to regulate the gene expression. So we have two functions for them, either housekeeping or regulatory. So for housekeeping, non-coding RNA, which will basically have more cellular functions, such as splicing. Splicing, by splicing we mean splicing introns and such on, or chemical modifications such as snRNA. For regulatory RNA, they will have more role into gene expression or gene inhibition, so, or activation. Example for that is microRNA or siRNA. So thank you everyone.